My name is Khalid Ahmed Al-Thilaya, a PhD student at College of Science and Engineering in Hamad bin Khalifa University in Qatar. In this video recording, I will present my PhD work titled An AI-based visual exploration framework for neuroscience and histopathology under the supervision of Dr. Jens Schneider. During the recent few years, clinical decisions have become heavily driven by medical imaging, which is a fundamental element of the clinical analysis pipeline. Most diagnostic tasks rely on a variety of scanning technologies to detect objects, providing evidence-based conclusions. Visualization of medical images is the key to extracting insight from images for a better understanding of various tasks, ranging from characterizing the relationship between imaging and diseases to neuroscientific research that seeks to study human brain structure and function. A shift towards developing sophisticated tools to exploit the large amount of information in these images has been rapidly increasing the recent few years. Several kinds of analysis tools have been developed to solve different kinds of problems in this context. Visual analysis and exploration solutions provide deeper insight into finer details of histology and neuroimaging data. These details carry a lot of important information that reveal clinical medical evidence without which correct and clear conclusion is hardly visible. Visual analysis solutions also facilitate some of the most challenging tasks in pathology and neuroscience by providing fast and clear visual exploration of data. In our research, we focus on three common visualization problems in digital pathology and neuroimaging. We propose a shape processing framework for visual exploration of cellular nuclear envelopes extracted from microscopic images arising in histology and neuroscience. The framework is based on a novel shape descriptor of closed contours. In the 2D case, it relies on a geodesically uniform resampling of discrete curves to compute unsigned curvatures at vertices and edges based on discrete differential geometry. The pipeline involves four main steps: extract closed contours, planar curvature to describe 2D shapes, circular parameterization, and elliptic Fourier analysis to form the feature vector. The input to the proposed pipeline is the set of segmented nuclear envelopes of cells obtained by applying a unit deep neural network on the images. Our descriptor is by design invariant under translation, rotation, and parameterization. We achieve the latter invariance under parameterization shifts by using elliptic Fourier analysis on the resulting curvature vectors. We further augment the proposed descriptor with feature coefficients obtained through sparse coding of the extracted cellular structures using key-sparse autoencoders. Our invariant descriptors provide an embedding into a fixed dimensional feature space that can be used as input features for deep and shallow learning techniques or as input for dimension reduction methods to provide a visual reference for clustering shape collections. We demonstrate the capabilities of our framework in the context of visual analysis and unsupervised classification of nuclei closed contours in histology and neuroscience images. We then extend the 2D shape processing pipeline for 3D reconstruction of neuroscience data by computing main curvatures based on the Laplace-Beltrami operator on triangular meshes, followed by computing a spherical parameterization through main curvature flow. Finally, we compute the spherical harmonics decomposition to obtain invariant energy coefficients. We demonstrate the capabilities of the extended framework in the context of visual analysis and unsupervised classification of 3D nuclear envelopes extracted from serial section electron microscopy stacks. We also propose an end-to-end framework for histopathology analysis of whole slide images. Our framework uses deep learning-based classification and localization of cell nuclei, followed by spatial data aggregation to propagate classes of sparsely distributed nuclei across the entire slide. We use YOLO model for localization instead of more costly segmentation approaches and show that using histogram boosts its performance. YOLO finds bounding boxes around nuclei at a good accuracy rate, but the classification accuracy can be improved by other methods. We extract patches around nuclei from the whole slide images and consider models from the SqueezeNet, ResNet, and EfficientNet families for classification. Where we do not achieve a clear separation between highest and second highest softmax activation of the classifier, we use YOLO's output as a secondary port. The result is a sparse annotation of the whole slide images, which we turn dense by using kernel density estimation, which generates a full vector of probabilities per pixel for each class of nucleus we consider. This allows us to visualize our results using both color coding and isocontouring, reducing visual clutter. Our novel nuclei-to-tissue coupling allows histopathologists to work at both the nucleus and tissue level, a feature appreciated by domain experts in a qualitative user study. We also propose a novel data structure called the mixture graph. This data structure allows us to compress, render, and query segmentation histograms of volume data. Such histograms arise when building a map of a volume containing segmentation IDs. Each voxel in the histogram map contains a convex combination of segmentation IDs. Each mixture represents the distribution of IDs in the respective voxel's children. Our method factorizes these mixtures into a series of linear interpolations between exactly two segmentation IDs. The result is represented as a directed acyclic graph whose nodes are topologically ordered. Pruning replicate nodes in the tree followed by compression allows us to store the resulting data structure efficiently. During the rendering, transfer functions are propagated from sources through the directed acyclic graph to allow for efficient pre-filtered rendering at interactive frame rates. We also apply the mixture graph to compute correctly pre-filtered volume lighting and to interactively explore segments based on shape, geometry, and orientation using multi-dimensional transfer functions. At the end, I would like to express my deep gratitude to my advisor, Dr. Jens Schneider, for his unlimited help and guidance during my PhD journey. I also would like to thank the Dean and Faculty of College of Science and Engineering in Hamad bin Khalifa University for their endless effort and support. I would like to thank the Islamic World Educational, Scientific and Cultural Organization and the Federation of the Universities of the Islamic World for organizing this competition.